My name is Laura Figueroa. This is Chapter 9, Listing Energy Sources for Skeletal Muscle Fiber Contraction. Muscle fiber contraction is a complex interaction of several cellular and chemical components. The sequence of events that results in the contraction of a muscle fiber begins with a signal from the motor neuron innervating that muscle fiber. The signal or neurotransmitter is acetylcholine, or ACH. The result is a movement within the myofibrils in which the filaments of actin and myosin slide past one another, shortening the sarcomeres. The source of energy that is used to power the muscle fiber contraction is adenosine triphosphate, or ATP. Now that we know what powers muscle fiber contractions, let's learn more about ATP. ATP supplies the energy for muscle contraction to take place. Muscle contraction does not occur without enough ATP. Muscles store a very low amount of ATP, only enough to power a few seconds worth of contractions. As ATP is broken down, it must be regenerated and replaced quickly to allow sustained contractions. There are three sources by which ATP can be regenerated, which are creatine phosphate metabolism, anaerobic glycolysis, and aerobic respiration. The first source of ATP muscles rely on is creatine phosphate metabolism. Creatine phosphate is a molecule that can store energy in its phosphate bonds. When a muscle is at rest, excess ATP will transfer its energy to creatine, which will produce adenosine diphosphate, or ADP, and creatine phosphate. Once a muscle starts to contract and need energy, creatine phosphate will transfer its phosphate back to ADP to create ATP and creatine. This creatine phosphate derived ATP can provide energy for muscle contraction at a very high rate, but only for about 8 to 10 seconds, at which point another energy source must be used. As the ATP produced by creatine phosphate is consumed, muscles turn to glycolysis as an ATP source. Glycolysis is an anaerobic or non-oxygen dependent process that breaks down glucose, also known as sugar, to produce ATP. Glycolysis cannot generate ATP as quickly as creatine phosphate. This results in a slower rate of ATP available to the muscle. The sugar used in glycolysis can be provided by blood glucose or by metabolizing glycogen that is stored in the muscle. The breakdown of one glucose molecule produces 2 ATP and 2 molecules of pyruvic acid. Pyruvic acid is converted to lactic acid, which allows the recycling of enzymes needed for glycolysis to continue. Glycolysis itself cannot be continued for very long, approximately 1 minute of muscle activity, but it is useful in facilitating short bursts of high intensity output. And the last source of ATP muscles use is aerobic respiration. Aerobic respiration is the breakdown of glucose in the presence of oxygen to produce carbon dioxide, water, and ATP. Most of the ATP required for resting or fairly active muscles is provided by aerobic respiration, which takes place in the mitochondria. The contributions for aerobic respiration include glucose circulating in the bloodstream, pyruvic acid, and fatty acids. Aerobic respiration produces approximately 36 ATPs per molecule of glucose, and it can supply ATP for several hours or longer. Listed here are the references that I used to obtain my information and pictures from. Thank you.